Museum in Berlin. So Wolfgang is a theoretical physicist like some of us, and it might interest you to know that during his PhD, he worked on traffic between cellular membranes, transport across cellular membranes. But today, he will tell us about how do we figure out what parts of a genome are under natural selection.
So thank you very much, Kavita, for the nice introduction and for the invitation to come to Bangalore. I really enjoy this meeting. So especially now, since I'm over my jet lag. I hear that some of you are physicists, so I have been a physics student and even a PhD student. So if you have questions, I have no problems. I understand your language. I can explain it. All right, so I moved from biophysics to evolutionary biology in the middle of the 80s. That was the time in Germany when there was a lot of discussion going on about ecology, about the environment. The Green Party arose in Germany, and so that was sort of a general movement. And I then decided I have to do something important in my life with what I had learned before. I had learned mathematics and physics. And so I moved to evolutionary biology, going to Sussex and to the US. I was 13 years in the US. And then there I learned what I'm talk about today. Okay, so the topic of my lecture is adaptation. As you may know that there are two main subjects in evolutionary biology. One is speciation. This is about how species are formed. And the other one is adaptation. I use a sort of a modern approach to adaptation. This is called population genomics. That means I look at the genome level to find the genes that are important in adaptation. Okay, and I'm trying to explain you the method how we found these genes, and then I also would like to use some data from my lab, from my previous lab at the LMU Munich, which we found through that method, and also show you how we can then functionally also get information that we are really dealing with a gene that we think we are dealing about. It's not only about theory, it's also some functional work, what we did. Okay, so much for my introduction. And the trait I'm mostly interested in, and when I started this project around 2000, was the adaptation of Drosophila melanogaster to temperate climates, yeah? Especially temperature adaptation. Drosophila moved out of Africa after the last ice age, so about 15,000 years ago. And my rationale was, while Drosophila migrated out of tropical Africa, there should be plenty of adaptations going on, especially concerning temperature adaptation, but also other adaptations. Okay, the method I'm describing now to find these genes that are involved in temperature adaptation is described is here. This is a simple model. Yeah, so from my heart, probably I'm a theoretical physics man, starting with a model and then trying to do something with it. Okay, and the model I'm using is so-called genetic hitchhiking, and this is a simple model of genetic hitchhiking. I start with a so-called neutral locus. This is a locus at which we have a polymorphism in a population. So there is one allele called capital A and one allele called little a. Okay, and This is supposedly neutral, and nearby there is a locus, a so-called selected locus, at which a beneficial mutation, here characterized by this blob, at some point arises. Yeah? Nearby means that the recombination between those two loci should not be too high. Yeah? Okay, so now let's see what happens after this beneficial mutation arose. Okay, so we see here two scenarios, that between this locus and this locus, there is no recombination. So recombination breaks up chromosomes and makes the evolution at this locus and this locus independent. Yeah? But if you have some recombination, or if you have no recombination, then what happens is, if this beneficial allele is going to fixation, that means the whole population, every individual in the population gets this allele. Yeah? At the beginning, it's only a mutant, but then it goes through the whole population. We say it is fixed. Yeah? If this is fixed, then you fix also this. Yeah? Because there is no recombination, so this allele has to do what this allele does. Okay, that's the reason why it's called hitchhiking. Yeah, this allele, so to speak, hitchhikes is this one. So if you drive from one town to the next town, and this hitchhikes along, this allele has to go where this is going. Yeah, so if this is going to fixation, this one has to go to fixation too. Yeah. And what the consequence of this is, if you fix this neutral allele, then this little allele disappears. Yeah, because all the individuals get this capital A allele. And what it means then is, at this neutral locus, variation disappears. Yeah, that's a very important point now. Yeah, because of this goes to fixation, at the neutral locus, you wipe out variation. And that is a reason why Andrew Barry called this process selective sweep. So sweep means you clean out variation. Yeah? Andrew Barry is now a philosopher of science, but while he was a student, he came up with this term, and we are still using it since 25 years. Okay, so selective sweeps is a word how we describe this cleaning of variation. Okay, this is this scenario. However, the other scenario is if we have some recombination happening between here and here, then it might be that the selected allele which appeared on this chromosome here will be recombining onto a chromosome that carries the little a allele at the neutral locus. Yeah? And that means this polymorphism will be maintained. Not in the same frequency, but it will be maintained. Okay. So what this is now, the consequence of this, and I would like to use this simple model to find these genes that are involved in adaptation. And why this is possible? Because we know that if we have selection going on, selection is driving adaptation. If you have beneficial mutations appearing, and if they go to fixation, such a allele will then determine adaptation. Yeah? So if a temperature-tolerant allele all of a sudden arises in a population outside Europe, this one is driven to fixation because it gives the fly an advantage. Yeah, that's the idea. Okay, so I'm studying it not the adaptation directly. I'm studying it through signatures at the genome. And the signatures are created by selection. And one of the signatures you see here, this is a selective sweep signature, you wipe out variation. Variation is plotted here, and here is a stretch of the chromosome, yeah, 40 kb, a 40,000 base pairs stretch, and we assume that the beneficial mutation happened in the middle of that stretch of DNA, okay? And exactly at this position where the beneficial allele arose, heterozygosity or variation is completely wiped out. Yeah? And if you go away from this side, from this selected side, then variation increases. And this data, what you see here, this is computer simulation data, these dots. And the curve is obtained by solving a diffusion problem. Yep. Speak up, please. Please wait, I can't understand.
I think I missed where you said the beneficial
The
mutation.
beneficial mutation happened here at 20 kb.
Okay. But how does that lead to increase in the variation?
Well, then the polymorphisms that are close to that selected site, they have no recombination, so they get wiped out completely. And the other one, which are a little bit further distant, they can recombine away. And those polymorphisms are maintained. But the frequency of the alleles which are initially linked to the beneficial allele is lowered. Okay. Other questions? So this is the principle. Basically, I look for these valleys of reduced variation in the genome.
All these sites have the same level of heterozygosity, is it?
The average heterozygosity is the same, yeah.
Okay.
It is about 0.01,
All
10%.
the 40 kb sites are
Yeah,
the same.
yeah. Of course, it varies, right? So it's neutral equilibrium level of heterozygosity. And this is then lowered here, and here it goes up to the original level. Okay, so another interesting phenomenon, another signature of selection is seen in the linkage disequilibrium data. So these are correlations between the polymorphisms in a certain genomic region. And the interesting thing is if you plot here linkage disequilibrium between a left locus L and a right locus R, these are neutral loci. So we have now slightly extended our original two-locus model to 3 loci, and you let then the selected locus here be here in the middle or maybe outside, it doesn't matter. You see the following picture of linkage disequilibrium, okay? So here is the time plotted forwards in time. At time 0, the beneficial allele popped up. And then it goes to fixation, okay? So this is this big figure. Here's time. It goes to fixation. To frequency 1. This is the selected allele. And while it goes to fixation, initially the linkage disequilibrium increases. Yeah? And at about frequency 1/2 of the selected allele, the linkage disequilibrium is maximum here at this position. Yeah? Okay. Those, I should say, here at this corner, the L and the R loci are located. Okay. And this S selected allele, it goes from left to right here. Okay. And here we have maximal linkage disequilibrium. Then after the time point when the level 1/2 is reached, and all of a sudden the LD drops. Okay, so such that if the selected allele is here in the middle, it drops down to 0. Yeah, that means across this selected site here, we see at the end of the phase here no linkage disequilibrium, but we see linkage disequilibrium on the left side and we see it on the right side. These are these M-shaped pattern. Okay, so this is a very interesting phenomenon because it can be used to localize where the selected allele occurred. Okay, now what I'm doing is I'm combining both information, linkage disequilibrium, and the reduction of variation, the frequency spectrum. And the reason is as follows. Both measures, the reduction of variation measured by lambda and the linkage disequilibrium measured by omega, they have a peak at the same site. Yeah? If the sites agree, then you have a good evidence that at that site, the genome, the selected allele occurred. Yeah? And I don't want to go into detail. This is important for non-equilibrium populations. That means populations that go through bottlenecks, for example. So now what my former student, and he really did the most important work here, already in 2002 it was published. Yusop Kim, he developed a test where he described the prediction of the linkage of the selective sweep model or the genetic hitchhiking model with the prediction of a demographic model. Yeah? So you have a neutral locus, a neutral model and a selected model, and you have to compare in a likelihood ratio test which of these models describes the data better. And he developed this test. Okay, he was the first one doing this, and then after we had this test, we started a genome search of Drosophila melanogaster. Yeah? So we sequenced, at that time there was no next generation sequencing, there was all Sanger sequencing. We sequenced short reads of the genome, 500, 600 base pairs, and went through about 300 or so sequencing reads per genome. And then we could see how much variation is there locally in these, along these chromosomes, and then could then spot those regions where the variation was reduced, yeah, with this test that Yusop Kim developed. Okay, and here I show you some data. The number of polymorphisms is in the left slide, and the frequency spectrum in the right slide. Okay, and so we looked at 3 different populations: one from Africa, where the flies are coming from, one from Europe, and one from Asia. Yeah. And the Asian sample was collected by Aparup Das. He's unfortunately not here today. I wanted to show him what came out of his collections. All right, and you see here on the left panel that variation, this is plotted here, is highest in Africa. This is clear because this is the ancestral population. Yeah, this is the same in humans. Africans are the most variable group of humans. Okay. Then you have the Asians. They are least variable, and the Europeans are not much more variable than the Asians. Okay, and we have data from the X chromosome and from the 3rd chromosome. Drosophila has only 4 chromosomes, really. Okay, and on the right panel, you see frequency spectra. And I talked about frequency spectra the other day. This is basically our most important source of information. And the frequency spectrum is quite different in those three populations. Here in Africa, you see that D is negative, while in Europe it's about 0. Okay, what that means? What is D? D is measuring the shift in the frequency spectrum, the deviation from the standard neutral model. Yeah? So we have neutral data. We assume no population size changes. If you have deviations from this in your whole data set, this is measured by D. And a negative D means is evidence for expansion, that the population size expanded in the past. That what D means. Okay? If you look at in a genome-wide level. If on the other hand, D, in this case, the Europeans and the Asians, they show no evidence for expansions. In this case, however, you see a variance that is too big according to the standard neutral model. Yeah? The variance here is smaller than in the non-African populations. And what this extended variance means is that these populations went through a bottleneck. Yeah? They colonized a new habitat outside Africa, yeah? Suffered from a reduction of population size, and then they increased again in population size to the size that they have now. So that the variance is increased in the bottleneck, yes. This is what you see here. It's exactly what you say. Yeah. And I can explain it tomorrow why that is. Okay. So here, this is evidence for bottlenecks. This is evidence for a more or less simple expansion. Okay. Now then, we did ABC analysis. This is sort of this is a method to analyze complex demographic models. It's more flexible than likelihood methods. And we had to distinguish two scenarios. Here is the African population, AF, and we look from the top down forward in time. So here is a past and here is a present. Okay. And in this model, the European population split off later than the Asian populations. This is one scenario, independently. And the other scenario is there was a split, one split from of the population from Africa here, and then later there was a split between the European and the Asian ones. Yeah. And let me briefly mention the result that is here. This model is much more likely. Yeah. So that means this is what you can imagine biologically reasonable. The ancestral population from Africa led to a population that migrated north, and this population may have settled somewhere in the Near East, where Israel or Egypt or so is nowadays. And this population then later split and gave rise to Europe and Asia. So this comes clearly out of this analysis. So you had a question. Yeah.
So did you use a specific, how do you come up with the standard neutral model?
How did I come up with this?
I mean, do you use a specific population to set up standard neutral model?
No, all we did was the parameters are free. They get estimated from this methods that we are using here. Yeah, you put in recombination parameters for Drosophila. Everything can vary, and then you estimate it. In this case, that is maximum likelihood. That's possible. Yeah. And we can even estimate where in the genome the selected allele popped up. Yeah, and how strong selection coefficients is and so. That can all be done in that estimation procedure that user can develop. Okay, so now we have a demographic model. Okay, we know that the European population went through a bottleneck. It split off from the Asian population, and we can also determine the times. That was about 5,000 years. That's about 15,000 years. Okay. Yeah, in order to simulate and do estimations, you use coalescence theory. Yeah, the Kingman-Coalescence. Okay, so we have now all the machinery now. We then used USOP's test, and we found candidate genes that show reduction of variation. That what we are looking for, that show a typical linkage disequilibrium pattern. And one of the first one we looked at is this gene. This is the fragment that we found is called wobble. So the fragment that we found in our screen had no variation in Europe. Yeah. Monomorphic, but there were several such fragments. We chose this fragment for further study because there were interesting genes in the neighborhood. And this one is a cytochrome P450 gene, which we thought is involved in environmental detoxifications. Yeah, of course, everybody would look at that. And there's another one, PGD. We thought it's also known that is involved in energy metabolism. Could also play a role in this adaptation. Okay, that's the reason why we chose this fragment. Okay, we could have chosen another one. So, and then what we did, we placed fragments for further sequencing around this monomorphic fragment. So here you see it, and the distance between these fragments is about 10 kb. We needed to see more. You know, that was just not good resolution before that. So we did all this, and what this came out. So here you see variation in Europe, in the European population, and here in the African population. And let's look at the African population first. Clearly, in these 12 additional fragments, they showed a valley of reduced variation. Yeah? That valley was about 15 kb wide. It did not go all the way down to 0, so there was some variation left. So that means the sweep, if this is one, is not very young. Yeah, but we can go with this USOP's test. We can look back about 50,000 years. We can see, you know, that's 50,000 years for Drosophila is fine. Okay, so how old this was? It turned out later it was about 50,000 years old. Okay, we could estimate this when this happened. Okay, and here is this wobble fragment. In Africa, the wobble fragment is not monomorphic. The wobble which was in Europe monomorphic. Okay. So this is the African one, 15 kb window or valley. In Europe, however, we see also a valley, but it is much bigger. Yeah. This is about 60 kb, and there was only one singleton which we found in this 60 kb, which is here. fragment 9 or 8, sorry. Okay, so this is what we got out of this. So then what we said, well, the European fragment is so big we cannot do much. Let's first look at the African one. This is the African valley of reduced variation. And then we sequenced everything what we could sequence in that region of about 20, it's a bit more, almost 30 kb. But we could not sequence everything. You see it here. There are some gaps because here, for example, this is duplicated gene. This is almost 100% identical with this one. So we were not sure. We could not interpret everything here. All right. So this is a gene called polyhomeotic. D stands for distal, and that polyhomeotic proximal. Okay. So as I said, almost 100% identical at the protein level. Oh, sorry. And the difference is here is a small intron, here is a large intron. Yeah? And this duplication, that is what we know from the genome sequencing of about 12 or 15 Drosophila species, is about 25 million years old. It's an interesting phenomenon that in 25 million years, there's almost no difference between here and here. And that what Bruce would immediately say, gene conversion, right? Here indeed there is evidence that at this position, there are the same polymorphisms. Yeah? And if you have the same polymorphisms in these two genes, you would have immediate evidence that there was gene conversion going on, which homogenizes these two genes. Yeah? How big are these genes? You see it's about 5 kb, these genes. Sorry? Yeah. We cannot say that. I think it must have been before, yeah. Or it could be that this polymorphism was copied over or from here to here. It could be.
I mean, I'm asking about the gene conversion hypothesis.
That I don't know, but yeah, we have too little data to say how much a conversion rate is and things like that. Okay. So the interesting thing is that although they are very similar at the DNA sequence level, that there is an intron, and just in that intron is a target of selection. Yeah? So this intron arose. This is pretty clear from the data of these 12 other Drosophila species arose after the duplication. And this, and a target, probably a regulatory site in that intron was the target of selection. And now you can make a nice molecular evolution story, because if really selection targeted a regulatory sequence in here, that is a possibility to cause functional divergence between these two genes. They are still, as I said, at the protein level very, very similar, but you see when you look at the expression profile, at the expression profiles, they start to diverge. Yeah? So this is probably a case of neo-functionalization. So this is a nice story. But my main story comes now. Look at this. This is this European valley of 60 kb reduced variation. What I said before, cannot do anything about it. But then I had a brave student, and she just did it. I don't know why she did it. Here is the diversity of the African population. Here is the 60 kb valley. almost no variation in Europe. And she sequenced everything around there. And then we ran, again, a test for selective sweeps. And look at this. This is a highly significant signature of a selective sweep in Europe. Okay, clearly part of this region, which is low in Africa, you see the African allele was just copied to Europe. Yeah, so the sweep in Africa happened before the African allele migrated to Europe. Okay, you can clearly see that, but this is only part of the story here. Yeah, the rest must have been happening outside Africa. Okay. And where did it happen? Well, from this profile, it's hard to say, but there is another signature of selective sweep, which I did not mention before, and this is what happens between population. Yeah? And the prediction is that strong positive selection drives divergence. Yeah, so strongly positive selected sites, you should find a higher FST compared to the rest of the genome. And there are several nice tests now, but we use Bayescan, it's a Bayesian method. And sure enough, it picked out these polymorphisms. They are significantly differentiated between Europe and Africa. Okay, so these are 5 polymorphisms that were picked out by Bayescan, nothing else. Okay, so where do they land in the genome? Well, they land in the intergenic region between PHP and this other gene, GC something. Okay, and then of course the rest is easy. Okay, you look at this region, which is about 5 kb, and check it. Well, now first let me look at this 5 kb region, and so clearly there is a European allele, there is an African allele. They are almost fixed. These 5 polymorphisms are fixed differences, almost fixed. You see it. There's a few deviations, maybe because these flies are collected near the airport in Harare, in Zimbabwe. Okay, so these are nearly fixed. These are nearly fixed, and in Asia, so Borneo and Kuala Lumpur, there is a recombinant between those two, the northern and the southern haplotype. Okay. Now, clear. Now you look. What is the molecular biology telling us here? Here are these 5 differences. And this region is known, this is a so-called PRE, which stands for polycomb response element. Yeah. That means this is a site in the genome, a region in the genome where the so-called polycomb group proteins bind to the DNA and regulate expression in the adjacent genes. What are the adjacent genes? Well, this is PHP and this one. Not in the other one, only in those 2. Okay. Now you can see how the story develops. It has been shown before by a French group, Fauvargue and Duras, that, and they checked it exactly at this, for this gene. Yeah. They showed that this regulation is temperature sensitive, and now you are in the temperature adaptation world. Okay, this regulation here, the silencing. So this is these polycomb group proteins silence expression. The silencing is less potent at lower temperature. Yeah, so the European ones silence, if this story, they should silence less than, because they are at lower temperature. Okay. So and then we set out, and again, this student, Susie Vogt, she made constructs. She combined the promoter of PHP, of this gene, and the promoter of this gene with the prey from Africa and the prey from Europe. Okay, and then she measured gene expression in those 4 constructs. Okay, and here she measured it on the whole flies, and then somebody said, well, you should look at some tissues. So she measured it for midgut and for another tissue. I think brain, there is no effect. But for the midgut, I'm not sure why midgut shows this. So in the Netherlands, in the European strain, where she measured it from the Netherlands, there is no difference, or almost, there's no significant difference in expression. Okay. However, and she measured it at 2 temperatures, 17 degrees and 28 degrees. Okay, and as I said before, in the Durand hypothesis is that the silencing is less potent in the lower temperature. Okay. So in other words, the lower temperature, this is a 17 degrees, shows a higher expression because the silencing doesn't work so well. And this is only in the African flies, not in Europe. Okay, and this is exactly the effect you're looking for. Because in Europe, apparently, they have adapted to this temperature, to these colder temperatures, such that the expression for low and for high temperature is about the same. And this is what we call canalization. Yeah? People think this canalization is important, otherwise the system doesn't work. You know, if the expression is temperature dependent and Drosophila have no other way to regulate temperature, they are really dependent on the ambient temperature. Something like that should be seen. Canalization should be an important trait. Otherwise, this adaptation to temperate zones would not work. Yeah. Okay, so this is my story, and in that gene, this is nicely seen. You see, this is significantly different, and also there's this difference between this and this. Yeah, so it's higher. But in the midgut, it's also the same direction, but it is not significant. Sorry, in that other gene, it's not significant. So that is where I am with my story. Now I have a few more minutes left. Now I'm retired. I've given up my lab. This was the last sort of experiment we wanted to do. This is what Susie did. And now you can see what we are doing now. And Kavita Chain spent a sabbatical in my group a year ago, and she solved this equation. She made a big attempt to this, and we would like to understand this equation. So what is this equation? Well, I have talked so far about selective sweeps and positive selection and things like that without having a phenotype. I didn't start from a phenotype. I started from selection at the level of DNA. And only later, after we had the gene, we talked about the phenotype. Okay. Now we would like to start from a phenotype and see what is a signature in this case? What is a signature, genomic signature in this case? Okay. And so Nick Barton suggested we should look at this model here. And this is a model, the so-called Wright equation, which comes from 1935. And the first part up to here, this is basically my equation that led to that fixation of the beneficial allele. Okay, but now here we have additional terms, and important are these terms here, because here is the phenotype, the mean phenotype in the population. Okay, and what is measured here is the distance of the mean phenotype from the optimum. Okay, and you see that this is important whether you see then a directional selection going on, and if the optimum is nearly reached, then this term kicks in and tries to reduce the variance of that trait in the population. So this is called stabilizing selection. Yeah? We can forget about this stuff here for the moment. This is only to get reasonable initial conditions. So now Kavita made a step at that, and to understand what is going on, and what she said, well, we have to look at two scenarios. We have to look whether the effects are mostly small or mostly large. Yeah? The effects are described by these gammas. Effect of a certain gene. Yeah? This effect tells you whether an allele at this locus makes you 5 centimeters bigger or smaller. This is, if you look at body size, that would be something like that. Okay. And this gives now a wonderful phenomena. You know, we try to understand them. What you still can see sometimes, such a fixation happens as in this simple case that I talked before. Yeah. So something like that also leads to a selective sweep. Okay. But this is a rare event, at least in this case where we have mostly small effects. Yeah? The small effects you see here, they do not much. They start from a frequency 1/2 and go up a little bit. Yeah. They do not much, at least the trajectories of these individual loci. However, those guys that are far away from the that are far away from, in the initial condition, are far away from the end. They make many contributions if you shift the optimum, the phenotypic optimum, to a larger number. Yeah. So that means, in other words, these guys, they contribute alleles that make you larger. Okay, closer to that optimum. To get closer to that optimum. Okay. However, and now the interesting thing happens is there is only one of those that goes to fixation. The other one, in the first phase, in the short-term phase, they all go in the same direction. Yeah? They all make you larger, but then all of a sudden they are stopped and have to go down again. You know? So this is a non-monotonic behavior of the red curve here, of this curve, and these effects are, this effect gamma 2 that has to go down. You see, so the weaker allele is weaker than the sweep allele. They go down, they are non-monotonic. Yeah? Something like that does not happen in that simple hitchhiking scenario. Okay, and now we are trying to understand what exactly makes them going down, but it is clear it is this term here. If you're close to the optimum, then all of a sudden the sign of this term starts from being positive to go negative. Yeah? And that leads then to a slowing down of this increase and eventually goes down to the stationary state. Okay, and the ones that have already plus alleles, that stay almost constant. So that's what we are trying to entertain now. These are the people who have worked on that. This is a theory group in my lab. You see here Yusuf Kim, who did really the most important contribution. Here's Kavita Chain. Here's Samasis Pavlitis, did a lot of work, for example, to distinguish demography from sweeps and so on. I talk about this tomorrow. And here are the experimentalists that did the Drosophila work. And here's the funding. This is a German funding agency. Then I had some money from the European Union. And these are some foundations that are also in Germany located. Thank you very much.
So in the last slide, I'm trying to understand why some of the alleles which have a lower fitness effect, like gamma 3 compared to gamma 2, and gamma 3 does make it sort of at high frequencies and gamma 2 gets lost.
Which one? Gamma 2?
2 and 3, if you compare 2 and 3.
2 and 3. Okay, 3 is a minor allele that has a completely different thing. It's interesting that this and this, so these are two major alleles, gamma 1 and gamma 2. And in this case,
How
we don't quite understand. I have to be honest, we don't quite understand. Here the major allele, the biggest one, makes it to fixation.
Right.
And the second biggest one goes down.
But it's not a major allele in the sense of frequency, right?
Sorry?
It's not a major allele in the sense of the frequency in the population, gamma 2.
No, the major effect allele.
Major effect allele, right. So I'm trying to understand between gamma 2 and 3.
Yeah.
3 has a much lower effect compared to gamma 2. Are these all coexisting at the same time? They are, right?
Sorry?
They are all coexisting at the same time at a given time slice.
Yeah.
So it's not independent simulations which are run.
No.
They're all at the same time.
This is a solution of the this model with 25 loci.
How the frequencies don't add up?
No, these are independent. I'm sorry. You're adding up.
The frequency go to 1.
Okay.
Yeah. And these minor alleles, they start as 1/2. This comes in through the mutation. There's a mutation selection balance at the beginning. We are at the optimum 0, and then there is a shift. Yeah.
In case, so the selective sweep happened in the African population. Selective sweep happened in the African population, and then the part of it formed the European population. If the particular area is not under strong selection anymore in the European population, we would expect new polymorphisms to arise in that region, right? But that doesn't happen. So can we assume from that, or it's still in selection?
So there is a sweep in the African population that happened about 50,000 years ago. Yeah. The African sweep, the swept allele migrates out of Africa, and then a second sweep happened at the nearby locus, one of these 5 polymorphisms, which we don't know which one. Preventing? Why that is not in Africa? Well, in Africa, you see this allele is not, the African version is monomorphic. There is almost no polymorphism in Africa. Yeah? And this, for some reason, came under positive selection outside Africa. Yeah? And then from there on, went to fixation in Europe. Yeah, that's the simplest scenario you can think of. Yeah? Because the environment changed there in Africa, there in outside Africa or in Northern Africa. Yeah, that's our best explanation. Okay, do you understand it?
So Wolfgang, I've actually seen something a little bit like this in a different setting. In the, I think the mid 80s, Russ Landy had a model of a major gene with background polygenes. And
that gave rise to this.
Yeah, it's a different context, but you see these, you know, in his he saw, for example, a major gene almost going to fixation and then slowly falling away as polygenes made up for the fact that, and so it's a different model, but it has a similar, it's addressing something different. You have these similar sort of readjustments.
Yes. So this Russ-Landy model, you have one major locus, and the rest of it is minor loci.
And the major locus has a deleterious effect as well.
Yeah. And this was the reason why. So Russ-Landy's model was picked up by a student in France, Miguel Chevin. I was in his committee, okay? And then he analyzed this model and claimed at a QDL, selective sweeps are not possible. Okay, that was published in Genetics in 2008. And then I said, I cannot be, you know, but I didn't find a mistake. Right? And then he used Russ-Landy's model, his formulation, and he could do something with it. It has only two differential equations, and so. Then I went home and said, okay, we have to have a real n-locus model. Okay, and then again we find the same thing. Sweeps are relatively rare at these QDLs, and here you see the same thing. You know, this is only one sweep, but if you go into a different area of the parameter range, what Kavita found out, she didn't find a sweep at all. Yeah, if you have mostly minor loci, right? And I know that I read Russ's paper. You know, I know these things. This is what we didn't discover that Russ detected that, and also Schema. We just try to understand. Sometimes this the largest effect allele goes to fixation, but in other parameter combinations, you have to wait. Maybe the fifth largest goes to fixation. Yeah? And the other ones have this behavior. Yeah? And it all depends on this slowing down of the increase in variation, of the increase of the frequency increase. So at the beginning, as I show you here, all the trajectories go upwards. Yeah. And then as soon as this thing here is close to the new optimum, then this gets smaller and then the other one kicks in. And then all of a sudden you have a change in the sign. Okay. Yeah, then it goes down.
I mean, the scaling looks like it depends upon gamma with respect to the displacement of the current population from the mean.
Yeah.
It's that current mean that's the thing that's driving all these weird dynamics. Because you can get there a lot of different ways.
Yeah. Yeah.
But it's a cool model. I mean, it's just, you know.
Sorry?
It's
a cool model. Oh, I love it. I love it. Okay. Yeah, there was, yeah. Sorry?
Range of values of gamma.
Say it again.
what is the range of the values of gamma?
Or it can be very small that, you know, some of these loci have almost no effect. You know, very, like body size. You have many loci that what Bruce also mentioned, and that could be very small, tiny effect on the, maybe on increase in body size. And some of them are near 1 or so. The important thing is what is the range of gamma with respect to the new optimum? That is the important thing. Yeah. If the optimum is changed to 2, then large gammas will lead to sweeps, to more sweeps. And if gamma is even bigger than that one, then you have a barrage of sweeps going through. Yeah. And what in nature, can you say something else? What is the effect size? I don't.
So they tend to be small. Building-based number one, most estimates people have an effect size of 6 feeding populations. And the problem is why a sample large effect on one tree probably affect a number of trees across the whole series. It's a effect. Those have big effects are rare. We sample the population trying to estimate effects by a sample, which you want to work on mutation. That's much of a turnaround.
So small is below 1, right?
Yeah.
That's what I have seen in the literature. Yeah. There was another question? Let's wait.
How the higher value of FST can be correlated with a strong positive selection? When you say higher FST value, we say that the population is highly structured. So how it can be correlated with positive selection?
Okay. So the FST, in this test, the FST is measured sort of genome-wide, yeah, between a whole set of populations. So we had a whole range of populations from Zimbabwe, even South Africa, then Uganda, Rwanda, Egypt, up to Sweden. Okay, so we measure FST, and then we look whether there's some outlier FST. And the idea is that if you have strong positive local adaptation, then you see fixations of an allele. Okay, and that can lead to this differentiation. Yeah. However, if you have balancing selection, the alleles in these local populations have about similar frequencies, and you don't see anything. Yeah? So with this test, you can distinguish between positive and negative, positive and balancing selection, basically. Yeah? That is going back to Lowentin and Krakauer's test. But that was heavily criticized. Coop does it with, he correlated with environmental variables. We have not done that.
We also met where you don't correlate. Also just
yeah. Other questions? Thank you.